Hey everyone, and welcome back to InfoPod. I'm your host, and really looking forward to today's deep dive.
Great to be here.
We're jumping into the really fascinating world of stem cells today.
Yeah, it's an incredible field.
These cells are they're quite remarkable, aren't they? This ability to make more of themselves, but also to change into other specialized cells.
Exactly. That dual capability is what makes them so fundamentally important. Self-renewal and differentiation.
Like the body's master builders, maybe?
Yeah.
Or a repair crew.
That's a good way to think about it. They're kind of undifferentiated, like a blank slate ready to become whatever's needed.
And that lets them, repair tissues and develop organs in the first place.
Precisely. They're crucial right from embryonic development all the way through life for maintenance and repair. They act as this sort of internal reservoir.
Okay, so they're not all the same then. You hear different terms thrown around.
No, definitely not. Their potential varies. We classify them based on, what they can become.
Right. So what are the main categories?
Well, at the very top, you've got totipotent stem cells. Think about the fertilized egg, the zygote.
Okay.
That single cell is totipotent. It has the potential to form absolutely everything, the entire organism, placenta included. It's the ultimate starting point.
Wow. Okay, the whole show. And then
Then you have pluripotent stem cells. These are maybe one step down, but still incredibly powerful. They can differentiate into any cell type found in the adult body.
so not the placenta, but muscles, nerves, liver cells, all that.
Exactly. All the specialized tissues. Embryonic stem cells are the classic example here. And that pluripotency is why they're so vital for research, you see. You can potentially generate any tissue type in the lab.
So if totipotent is building the whole universe, pluripotent is building any planet within it.
I like that analogy. Yeah, something like that. Then we move to multipotent stem cells.
Okay, multipotent.
These are more restricted. They can still form multiple cell types, but usually within a specific lineage or tissue family.
Like blood cells.
Perfect example. Hematopoietic stem cells, or HSCs, are multipotent. They live in the bone marrow and generate all the different kinds of blood cells, red cells, white cells, platelets, everything.
Got it. So still versatile, but within a certain job description.
Exactly. And finally, you have unipotent stem cells. These are the real specialists.
Just one thing.
Yep. They can typically only produce one specific type of cell, but crucially, they can still self-renew, making more of themselves. Think of certain skin stem cells, maybe.
Okay, that makes sense. Totipotent, pluripotent, multipotent, unipotent. Different levels of potential.
Precisely.
So let's circle back to those pluripotent ones. The embryonic stem cells, or ES cells, they sound like they have enormous therapeutic potential. Where do they come from again?
They're derived from the inner cell mass of a blastocyst, which is a very early stage embryo, just a few days after fertilization.
And scientists can grow these in the lab.
Yes, and that was a major breakthrough, developing methods to culture them, to keep them dividing, and maintain that pluripotent state indefinitely. This created human ES cell lines.
Which are used for
tons of research, studying early human development, understanding how diseases start, and crucially, exploring ways to generate specific cells for therapies.
Right. This is where it gets really exciting, the idea of making new cells to replace damaged ones.
Absolutely. If we can figure out the signals, the instructions that tell an ES cell to become, say, a heart muscle cell or a neuron.
Or an insulin-producing cell for diabetes.
Exactly. That's the goal for so many degenerative conditions. Parkinson's disease, Alzheimer's, type 1 diabetes, heart failure after a heart attack. The list goes on. The hope is to regenerate tissues that are damaged or lost.
It really does sound almost like science fiction becoming reality.
It's incredibly promising, the potential to grow specific cells on demand. Imagine tailored skin grafts for burn victims, functional liver cells for liver failure, maybe even repairing spinal cord injuries someday.
Well, it's not quite there yet. There are challenges.
Oh, definitely. Significant challenges remain. There are ethical considerations surrounding the source of ES cells, potential immunological issues like the recipient's body rejecting the transplanted cells, and still many technical hurdles in controlling differentiation perfectly and ensuring safety.
Right. Important caveats. Okay, let's shift gears a bit. You mentioned hematopoietic stem cells, HSCs, earlier. The multipotent ones in bone marrow, are they actually being used now?
Yes, absolutely. HSC transplantation is a well-established standard therapy. It's been used for decades, actually.
For what kinds of condition?
Primarily three main areas. First, treating inherited immunodeficiencies. like severe combined immunodeficiency, SCID, sometimes called bubble boy disease. A transplant can basically give the patient a whole new functional immune system.
Wow. Okay, what else?
Second, correcting genetic blood disorders. Think sickle cell anemia or beta thalassemia. Replacing the faulty HSCs with healthy ones can produce normal blood cells, offering a potential cure.
A cure. That's huge.
It can be, yes. And third, it's used as a support therapy after very high-dose chemotherapy or radiation for certain cancers.
Because the cancer treatment wipes out the patient's own bone marrow.
Exactly. It destroys the cancer cells, but also the healthy HSCs, so the transplant restores the blood-forming system afterwards.
That's incredible that this is already happening. Are there specific advantages to using HSCs?
Yes, several practical ones. A big one is their ability to home to the bone marrow.
Homing.
Yeah, you just infuse the cells intravenously into the bloodstream, and they naturally find their way to the bone marrow and start working. No need to surgically implant them.
That's amazing. Like they have GPS.
Sort of. Also, you often only need a relatively small number of donor HSCs to completely repopulate the recipient's bone marrow, and they can be cryopreserved frozen for long periods, which helps with logistics.
Okay, so how do doctors actually find and collect just these HSCs? You said they're only about 1% of marrow cells.
Right. They use markers on the cell surface. The key one is a protein called CD34. It's like a little flag on the HSCs.
So they look for the flag.
Pretty much. They use antibodies that specifically stick to CD34. These antibodies can be attached to tiny magnetic beads or used in cell sorting machines. This lets them pull out or select the CD34 positive cells.
Enriching the stem cell population.
Exactly. Concentrating the HSCs and getting rid of many other cells, this CD34 selection is common.
And you use this for different types of transplants.
Yes, both autologous, using the patient's own cells, and allogeneic, using a donor's cells. A major benefit, especially in allogeneic transplants, is that selecting for CD34 plus cells helps remove contaminating T cells from the donor graft.
T cells. Those are immune cells, right? Why remove them?
Because those donor T cells can cause a serious complication called graft versus host disease, or GVHD.
Okay, let's unpack those transplant types. Autologous, allogeneic, what's the difference? And syngeneic.
Right. So autologous means self. You harvest the patient's own HSCs, freeze them, give the high-dose therapy, then thaw and return their own cells.
Advantage. No rejection.
Correct. The body recognizes the cells as its own. But you can't use it for genetic diseases, obviously, because the patient's cells have the defect. And maybe not for some cancers if there's a risk the harvested cells are contaminated with cancer.
Makes sense.
Syngeneic, that's when the donor is an identical twin. Genetically identical. So again, very minimal risk of rejection or that GVHD problem. But of course, you need an identical twin.
Which is rare. So the most common must be allogeneic.
Yes. Allo means other. The donor is genetically different, could be a matched sibling or an unrelated matched donor found through registries.
And this is where the immune complications come in.
Precisely. Two main risks. One is host versus graft disease, where the recipient's immune system, if it hasn't been fully wiped out, attacks the donor cells. The bigger concern usually is graft versus host disease, GVHD.
Which you mentioned is the donor T cells attacking the recipient.
Exactly. The transplanted immune cells, particularly T cells, see the recipient's tissues, skin, liver, gut, are common targets as foreign, and they attack.
That sounds serious. How is it managed?
With immunosuppressive drugs. These dampen the immune response to prevent or treat GVHD. But the downside is they also weaken the immune system overall, increasing the risk of infections and potentially even secondary cancers later on. It's a balancing act.
Definitely complex. So where do doctors get these HSCs from? You said bone marrow. Are there other sources?
Yep, 3 main sources today. Bone marrow is the traditional one. It involves aspiration, usually from the donor's hip bone under anesthesia.
Okay, well.
Peripheral blood is now very common. Donors receive injections of a growth factor called GCSF for several days.
What does that do?
It mobilizes the HSCs, kind of encourages them to leave the bone marrow and enter the bloodstream in large numbers.
Ah, so you can collect them from the blood.
Exactly. Through a process called leukapheresis, it's like donating plasma blood is drawn, the stem cells are filtered out by a machine, and the rest of the blood is returned to the donor. It avoids general anesthesia.
Convenient. And the third source?
Umbilical cord blood, collected from the placenta and umbilical cord right after a baby is born. It's rich in HSCs.
And any advantages there?
Well, the volume is smaller, so it's often used more for children or smaller adults. But a key advantage is that the immune cells in cord blood are less mature.
Meaning less risk of GVHD.
Generally, yes. The risk or severity of GVHD tends to be lower with cord blood transplants, which is a significant benefit.
Interesting trade-offs between the sources. Okay, one more really exciting area, combining HSCs with gene therapy. How does that work?
It's a really powerful combination. Because HSCs self-renew, if you can correct a genetic defect in the stem cell, that correction should last for the patient's lifetime.
So you've fixed the source.
Essentially, yes. The basic process is you take the patient's own HSCs out. Then in the lab ex vivo, you use tools, often specially engineered viruses, to deliver a correct copy of the faulty gene into those cells.
So you're genetically modifying their own stem cells.
Precisely. Then you infuse these corrected cells back into the patient. Since they're the patient's own cells, there's no risk of rejection or GVHD.
And they take root in the bone marrow.
Yes, they engraft and start producing blood cells that now have the corrected gene, and because HSCs self-renew, they keep doing it indefinitely.
That sounds like a true cure for some genetic diseases.
That's the promise. It's showing amazing results for conditions like SCID, those kids born without an immune system, and holds huge potential for sickle cell anemia, beta thalassemia, and various other inherited disorders. It avoids all the immune complications of allogeneic transplant.
Just incredible. The pace of progress seems relentless.
It really is. When you look at the whole picture, from understanding the basic biology of these cells, their different potentials,
To the established therapies like HSC transplants, and now the cutting edge of gene therapy and the future promise of pluripotent cells.
Exactly. It spans from fundamental development to potentially revolutionary treatments for a vast range of conditions. There's still a way to go for many applications, but the trajectory is undeniable.
It truly underscores the remarkable nature of these cells. Okay, well, that feels like a good place to wrap up our deep dive for today. As the research keeps moving forward, it really does feel like stem cells offer immense hope for changing medicine.
No question about it.
What future breakthroughs do you think are maybe the most compelling to watch out for? Definitely food for thought for everyone listening.
Lots to consider there.
And a huge thank you for joining us on InfoPod today and sharing all that insight.
My pleasure.
And to all of you listening, if you found this journey into stem cells interesting, please do give us a like, maybe leave a comment with your thoughts, and share it with anyone else who might be curious.
Yes, spread the knowledge.
And of course, hit that subscribe button so you don't miss our next deep dive. Until then, thanks for tuning in.